Do you remember? You're going to hear this a lot again today. Antigen presenting cells. Okay. So do you remember there's three main kinds? There's what? The macrophages are great at doing that. Do you remember the dendritic cells are great at doing that? And B cells can do it too. And they can kind of present to other B cells and so on. Okay. So what do we have first? Well, even in your innate system, remember the innate system is keeping everything hopefully down, bombing it and doing fever and, you know, eating things, but they're also picking up information. And so as soon as you start eating foreign cells and chopping them up, we call it processing, and putting it on the sticky outside, remember on that MHC complex, they'll start producing cytokines. Now, there are many cytokines, and again, you'll hear a lot of them again and again, and even next unit, you're going to hear them again. They're very important for communication. And interleukin-1 is great. It gets everything going. And so pretty soon, you know, you get more leukocytes, you get lymphocytes, you get these helper T cells. And those helper T cells bind to the antigen presenting cell. If the antigen presenting cell is presenting, then the helper binds as well. And it's all what? The MHC, that complex, everybody's kind of bound up together. And then we start producing the other interleukins. Can you see interleukin 2? And then we get, in addition, that helps to spark off a third type of cell. Now, we're talking about not T cells right now, we'll get to them, but we could trigger Bs and Ts at this point. But what are we going to focus on? The Bs, right? But it's the same principle. If you're a matchy match, in you come and you click in again. So there's a lot of presenting and then everybody clicking together, kind of like a seatbelt in your car. You know how the seatbelt clicks in? That's kind of how I see this whole process. And as soon as you get a B cell binding, it gets activated. What do we mean by activated? We mean it starts to do the P word. Remember the P word is proliferate. And immediately we get two types. So there's what? The shooters, right? They are so hardworking, they don't last long. They're what we call short-lived. But don't worry, more are being made, more are being made, more are being made while the infection is going on. And then for next time, we have memory B cells, which we need to talk about in a minute. They're the more long-lived. And in fact, the long-lived B cells, you hope that they're here in your body for a long time, ready. They're ready for action any time. And that can be for the next 70 years if you're lucky. Now, unfortunately, as we get older, they fade a bit. And that's why often if you're getting vaccination, don't we give you boosters at certain ages? Because we want to maintain our memories, because you are a walking history book of every disease you've ever had. And you don't want to get that disease again. And your body will do everything to try and stave it off. So are cytokines important? Yes. And we've got our B cells getting going here. All right, and then remember I said it's like everybody clicks in together. Everybody's clicking in, kind of like seatbelt style. Everybody's clicking in. You see the touchy-feely stuff going on here? Pretty much, again, reminds me of a seatbelt clicking into the, you know, the little pocket in your car. So, why do we want clonal expansion? Well, the more, the better of each clone, because what are they going to do? They're going to shoot those plasma B cells. Remember, they're going to shoot an awful lot of antibodies into the fluids, you know, wherever the injury is, in the blood, in all the humors, we call them, the liquids in your body. And so that's for now. This is for later. Now, the cool thing is, if you're still alive and you make it through this part, then for the rest of your life, you have the ability to do a rapid response to what we call the second exposure, the third exposure, the 20th exposure, because you are always being exposed to pathogens. We can't get away from them. That's part of being human. Yay! Oh, my cat's killed a mouse this morning in my house. Oh, I know, I'm like, oh, I know. And then I'm like, but you didn't eat it, thank goodness. Okay, so memory cells. Let's talk about antibodies. It's one of those mornings this morning. Oh dear. Antibodies. Guess what they're made of, everybody? Protein. Remember, everything important is protein. So they are proteins that are released by plasma B cells. Remember, they're the ones that shoot them out. They also have them around. Remember, every B cell is coated with them as well. Remember, they're in place, stationary, as we call them markers or receptors, and they can shoot loads of them. So they bind very specifically to that part of the antigen that we call an epitope. Remember, the subunits of an antigen are called an epitope. And if we look at let's look at our typical antibody. It's drawn out as a diagram here. Can you see there's a short chain of amino acids? We call it a polypeptide. Can you see there's a longer chain? And then mirror opposite, same duplicated. Can you see the longer chain and the lighter chain? So there are how many chains? Well, 4, right? 4 chains, so 4 polypeptide chains. And we call the short one the light one because it weighs less. And we call the long one the heavy one because it weighs more. So that's the way it's named. And as you can see, they're held together. Can you see an S and an S? So 2 S's, they're called disulfide bonds. Very important for stabilizing proteins, as you know. So if we look at our antibody, can you see we have it's like it reminds me of a little flower, like a tulip or something. And you see it's got like the stem of the flower. And so that's the bit that actually connects. You know when I said that B cells are all coated with them? This is the bit that plugs in to the plasma membrane. So it's kind of like it's like almost like a furry with antibodies sticking out. But where are the tips? Well, can you see there's one tip here that binds the antibody and one tip that binds here. So what is going to stay the same? What's going to be uniform? Well, the bit that's part of us, right? The human part, the part that's going to plug into our membranes, the stem region. But what's going to vary because of all the genetic shuffling? What we call the tips. We call them V. V means variable, yeah? Now, if you look at just one of these variable tips, can you see, I mean, this is drawn as a box. It's not very realistic. But can you see, can you see all the little amino acid bubbles there? Yeah? Mm-hmm. And can you see this, it's got a groove. Like all proteins, they have a groove. So it's like a little groove there. But that groove is going to be different on every flavor of antibody. And that means, can you see the little epitope that's come from the foreign pathogen? Can you see how it fits? So it's very much like an enzyme and a substrate. You know, that's a protein thing, isn't it? And so it will connect how many? Well, can you see one pathogen epitope here and one here? So technically it can tie up 2 pathogens, right? In theory.
Antigens are pathogens, right?
2 pathogens. Pathogens are built of foreign molecules. Remember, we call them pampers. Mm-hmm. Pathogen-associated molecular patterns, right? And receptors, pampers. And those pampers are foreign. And so our body recognizes them as foreign and they generate our response. We call it the foreign bits antigens. Okay. They have to be big and they have to be protein in general. Remember we went through that, the 2 things, big and more protein like the better, yeah? Well, now we're on the antibodies and guess what? They're protein as well. And they have to, they're always made of protein. And they will bind to the bits, the foreign bits, the foreign chemicals, and they fit like a key in a lock. So cool. All right, so if we look at it in a bit more detail now, then what did we just say? We said, yep, it's got the 4 polypeptide chains with the, what did we say, the heavy and the light. We've got, what did we say, variable tips. Yeah, that's what makes one antibody different than another, is the tips, the little grooves. And they are, because of that, they are very specific, like an enzyme is to its substrate. Nothing you don't know already. Now then, what did we say about the stem region? That's the constant, because it's human, right? We're humans, we produce human antibodies. And that can bind to your B cells. Remember, to your cells, to you. So that's constant. And that's how come it can be a receptor, because what does it do? It plugs in and it's actually sitting as a receptor on the membrane. Right, now the other thing I want you to notice, and by the way, this is other drawings of the same antibody. This is the same thing in different textbooks. I always like to show you different views. That one looks like peanuts to me. Not sure about that one. This is probably the more realistic representation, because you see all the bobbles in the peptide chain, you know, and it's all globular, isn't it? And have you noticed the name? Can you see it's called a globular protein? Can you see the word globulin in there? Which means it's got primary structure, secondary, tertiary, meaning there are many turns and spirals around each other. And it's also got more than one polypeptide chain, right? Which is globular, quaternary structure. And then is it a defense molecule? It is. Your immune system is your defense system. So everything's named very logically, immunoglobulins. And can you see how we shorten it? Because it's a bit of a mouthful going, immunoglobulin A, immunoglobulin G, immunoglobulin F, whatever. So we just write IG, immunoglobulin. And you'll see that from now on. So one more thing to point out is, can you see, to me they look like a pair of knees. You know, if you're an antibody, right, and you've got what, your stem area, right, and then you've got your, okay, well I'd have to be 4 arms here, but you know what I mean, 2 on this side and 2 on this side. Then at my knees, something magical happens if I find an antigen to bind to. My knees trigger complement. So do you remember, let's review complement. Complement consists of more than 20 different proteins, of which there are trillions floating around in your liquids of your blood, right? Trillions of them. And as soon as you trigger one, it's like the horns of a cow, remember? If you trigger it, and there's multiple ways to trigger it. Remember, one way is just being a pamper. You know, if you've got endotoxic, you know, you've got a gram-negative wall, that's a pamper. You know, if you've got funky things on there, that's a pamper. So that's called the alternative pathway. Do you remember that's the one where it just, anything that's a bit foreign gets triggers, right? We call that the innate or second level. Remember we talk about the first level of defenses, the second level. Now I'm telling you, complement is coming back again, because you can get complement get going even more, because as you're binding, antibodies are what? Going to find their target? That's like marking them, death, right? So who's going to come along and kill the pathogen? Remember how complement works? You trigger it, it's like those dominoes, right? And at the end of the pathway, you get these little proteins that will start knifing whatever pathogen this is bonded to. So that's kind of cool, isn't it? So wherever it binds, watch out. It's marking it for death. Yeah. So, can some immunoglobulins bind complement? Yes. Where? Can you see the little kind of little areas? Kind of in that stem region. Kind of like the knees, I would say. Okie doke. So what are the different types? We have 5 major types of IGs, immunoglobulins. Can you see the 5 flavors? Can you see there's IgG? Can you see we've got IgAs? They're often in pairs. Do you see the 2 together? Can you see we've got IgDs, IgEs, and then my favorite one is Big Mama, IgMs. Why do I call it Big Mama? Well, how many pathogens in theory could this one bind? 10. If you can count, you can tell me. If there's 5 of them strung together and each one has how many binding sites?
2.
What's 2 times 5, everyone?
10.
Yeah, there we go. So, okay, so say you've been invaded and you're trying to keep a lid on things. You do not want that pathogen to become systemic and start going through to your liver, your kidneys, your brain, your lungs. You want to keep it in an area. So when B cells start shooting, they change which antibodies they shoot as an infection progresses. What would you want to shoot at the beginning of an infection? One that ties up 2 pathogens? Or one that ties up more? So which one do you think your body shoots first?
IgMs.
You're correct, well done. We can use that diagnostically. So, say we think you've got red measles. To be careful, when you write measles, it's meaningless. Measles, there's multiple types of measles. And so again, Appendix D, I'm giving you a bit of a nag here. Make sure you know kind of which measles it is. Because isn't there red and German as well? In other words, and red measles is rubeola. Don't get them confused. German is rubella. Slightly different spelling, but oh my gosh, whole different disease. Okay, let's say you have a rash and they're trying to figure out what's wrong with you. And they find antibodies to rubeola, the red measles. It makes a difference which kind of antibodies. Because they can do a titer. Remember, titer means how many have you got? And if they look at your titer and you've got a heck of a lot of these, that means you're sick now. But if you have the other ones, it could be that you had measles, red measles, when you were like 10 years old. But you just have the, remember you're a walking history? Yeah? Okay? So that's a diagnostic. You'll hear that again and again, but Unit 4 is about diagnosis. You know, we get to put everything together and talk about diseases, Unit 4. You'll like that. So what's different is the stem and how they're organized. Let's talk about those different antibodies now. So IgG are the commonest, and then after that, well, IgMs are the third commonest in general if we're doing a titer, if we're doing a differential titer on your blood. You kind of need to come up with a mnemonic, a bit like, you know, when we were saying about the frequency of leukocytes. Which antibodies, what's the frequency? I have a really pathetic, well, I don't even like this mnemonic, because I don't remember it as well as I should. If it were ruder, I would. But maybe you can come up with something better. My mnemonic is give, A, monkey, daily, exercise. Give, A, monkey, because we're on monkeys, aren't we? Give a monkey, daily, exercise. Now, if you want to come up with something ruder that's yours, do it. Because that helps you remember kind of the overall holistic frequency of antibodies of all diseases that are currently in your blood right now, okay? So if we focus, let's start off with the, what was it? Give. So that's our what? IG.
A. Oh, G.
Give.
Sorry.
Give is G.
So sorry.
Oh, it's all right. And then are we going to pass the roll sheet around, darling? Would you mind? Go ahead and sign in and pass it around. Thank you. Okay. So if we look at IgGs, they are definitely in the majority. Percentage-wise, 80%, everyone. 80%, that's a lot, right? And so what's cool about them is they're the only antibody that can get to your unborn baby. So they can go across your placental barrier, and since they can get into the fetus, that means when you're born, you're going to have mom's IgGs. And if you're an embryo, you'll have mom's IgGs. Everything mom has, you get. So I know people were very concerned about, for example, getting a vaccine for, you know, in the COVID pandemic. They were concerned if they were pregnant they were worried about the vaccine. Well, it turned out it reduced the likelihood of fever and therefore fewer abnormalities and a higher birth weight. So there are these papers coming out now because we don't know, do we, until we analyze the data. But it turned out all those babies were getting protected because mom was forming antibodies and baby got them too. So that's kind of nice. So what do IgGs do? They do everything. Now, you know that when an antibody binds, right, when an antibody binds, it binds where? By the tips? Isn't it from the tips? And so if you bind an antibody, and I'll see if I can get to the thing there. So if you have a pathogen, right, there's a pathogen, and if you have an antibody that binds, can you see what's kind of sticking out? Can you see? Which part of the antibody is sticking out?
The stem.
Okay. And the stem is really good at binding to us, right? So the blank is they enhance phagocytosis. So let's try and figure out why. How come IgGs binding to the pathogen will enhance phagocytosis. Well, let's think about that analogy we used of a slippery bar of soap right in the shower. If you drop it, remember, if you're a phagocyte, you've got to be able to what? Pick it up? And it's slimy, right? Because it may have a capsule. So by having antibodies bind, can you see it's very similar to complement, and that what you're doing is, it's like shooting antibody nails into it. So now, can the phagocyte now attach more easily and pick it up? And if it can attach, it can now process it better and destroy it, right? And so, what's that process whereby you coat a pathogen with sticky out bits?
Opsonization.
It begins with an O, that's right, opsonization. And opsonization, in fact, I always think about that Korean singer who goes, op, 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 Gangnam style. You know, the op, Gangnam style is coating everything. Okay, don't listen to me. So we got that. So can you see why that would enhance? Because now the phagocyte can come along and go, yeah, and like do the phagocyte because it's got something to hold on to. Yeah. So opsonization. Okay, everyone say opsonization.
Opsonization.
Okay, so complement can do it, but remember there's more than one way to get things going, to coat. So remember, complement was those proteins that can act like what? Like javelins or I don't know, swords or whatever, like little nails, and coat. So opsonization, that's right. Remember they have knees. Remember the knees? When it binds to the pathogen, the knees of each of these, remember they have a little sticky out knees? Do you remember that complement can go in now and gets triggered? Remember that's like the bulls of the horn? So all of these guys are triggering complement. And you know there's trillions of complement molecules around, and they just go, wah, and off they go. And then, MAC attacks, remember that? You know, the C9, the C8, C7, they form these rings, you get MAC attacks. And so, so antibodies. That's just a couple of things. What else do they do? IgGs are fabulous for neutralizing poison. Remember, poisons are called toxins? And they can neutralize it because normally a poison would what? Go to your, I don't know, neuromuscular junctions or you know, to protein synthesis or something like that, right? Or to your blood. And by binding, that poison can't work. So it neutralizes the poisons, the toxins. And we find massive numbers of IgGs in the second response, you know, the second time. Because remember, what are we going to see the first time is going to be these guys, which I'm going to talk about next. Okay? So we find them massive production if you ever get hit by that pathogen second time. And so, since that's kind of memory, isn't it? Second time, third time, fourth time, no wonder they're a very important part of long-term immunity. So, you know, when you're 80 years old, you hope you have lots of IgGs, right? Okay, let's talk IgMs. They're not the commonest, not even the third commonest, but they're important. About 6% roughly, and I'm not worried so much about the percentages, as remember what I've said, the kind of the order. And so they're pentamers, as we already said, there's 5 of them stuck together. And because they are big, they are limited to mainly being shot out and circulated in your blood vessels, because they're just too big. And they can be receptors on the outside of the B cells as well to give it more kind of binding power. And the big deal about IgMs is they are the first antibodies that we'll see in you when you're sick against that pathogen that you're sick with now. And that's because they are great at tying them up. What are we posh name for tying up? Cross-linking? Remember we talked about agglutination? Any A word there. Agglutination, aggregation, any A word that means to clump, basically. So they're good at clumping, cross-linking, agglutination. And if you think IgGs are good at complement, you got a whole bunch of knees there, don't you? And so they will activate complement. So they're the first to appear in an infection, and that will be significant when we talk about diagnosis. They're fairly, because they're proteins and they're easily damaged, then they don't last a very long time, short time. And what did we say? If we find them, then the patient having high levels means they have a primary infection with the disease agent that we found. But they, IgMs. So if you have IgMs to, what do we say, to red measles? You got measles now, right? All right, IgAs are the second one in line. And IgAs, they're about, 13% roughly, 13%. And they're in dimers. Remember, they're the twos together. And they protect your mucosal cells. So, you have a lot of lining, through your gut, through your mouth, all sorts of orifices. And so they're very good at preventing attachment. Because think about it, in your food, you'll have pathogens trying to attach to your gut. And by having these IgAs, they'll prevent attachment of bacteria and viruses. Now, what's cool about IgAs is, again, we said when you're a mom, remember you give IgGs to your baby, but not to your newborn. You give it to your fetus and embryo, right, across the placenta. But now the baby's cut off because the baby's born. But one of the reasons why we do encourage breastfeeding is because of the colostrum is very rich in antibodies. Which ones? These. So these are the only antibodies found in breast milk. And one way to remember that is what you say when you see a baby. Aww. Okay, maybe you don't like babies. Okay, kitten, good kitten. Maybe that helps. To puppy. Okay, so what they're getting is IG. What does a baby say when it sees a nipple and it's hungry? Still, same thing. Okay, very good. Okay, IgDs, 1%. Can you see we're getting really low now? 1%. And so they're the ones that are very important for getting things going. We find them on B cells. Remember, B cells are coated, and so the receptors are often IgDs that are going to be clicking in. Remember getting everything going in the beginning, the original kind of clicking in and interacting with the phagocytes and interacting with the pathogen, often IgDs, and they will stimulate B cell activation. So they're good for getting things going, okay? So IgDs, yep, getting the job done, right? Igs, what do you see? You know how at the moment we've got lots of allergies? Oh my God, poor Dr. Greg Russell, he's like streaming. He's awful at the moment. And he's taking like 2 Zyrtex, a couple of other drugs, and he's like streaming, because allergies, what do you get? Lots of snot and mucus. And what do you say when you see a worm wriggling out of your butt? Ew. Ew. And when you have lots of stringy snot coming out of your mouth and out of your nose. Ew, right? Okay. So IgEs are the lowest of all. Oh my God, how low can we go? 0.002%. That's low. Because they're tied in place. They're on your mast cells. Mast cells? Tissues close to the surface, mast cells. They're a bit larger than the IgGs. And they're mast cells. Also, one of the films that we find patrolling superficially are your basophils. Okay? And they're very common, therefore, in close to the surface. So that we've got your mucous membranes. And they're the ones that make you your eyes drip and your nose drip in allergic reactions. Sometimes we have that in the gut and you have more diarrhea. When you're having, some people have allergies that way. So they participate in allergic reactions, and they're part of what we call the innate response. Remember when you get injured, where is it going to be? Superficially, right? And remember how all the cells shout? Okay, these guys are involved. Okay, so there are antibodies there as well. Okay, so what the heck do they do if they make you feel so miserable? Let's talk about the good side. I started on the bad side, didn't I? Let's talk about what they do that's good. They help to protect your body against parasites. So what are the main parasites? Protozoans and worms. Again, IG. Ew. Okay, stop them there. Yeah. Very good.
Yeah.
Okay, so, you know, you can make a little chart of that if you want, and that's kind of cool. I skip over that because that's just repetition. Okay, so everybody, let's just look at this from another angle. Everything we've just talked about. So can you see time on the bottom? Can you see on the top how many antibodies do you have to a particular specific pathogen? Well, first, what's got to happen is the pathogen has to get in, right? Pathogen gets past first line of defenses. Hopefully, second line of defenses are operating now, otherwise you die, right? Because how long does it take before? Can you see what happens when we get the number of antibodies going up? Check it out. That's a long time. That's what, 10 days? In the meantime, what are you doing? Fever, complement, inflammation, all that good stuff, right? Getting everything going. Phagocytosis, woohoo! And information gathering. So what do we get? Well, we're talking about the B cells. So who's going to be activated? Who's going to be proliferated while we're looking at this? The B cells. The B cells, remember, there's the ones that are for now and the ones that are for later. Remember, the ones for now are called plasma B cells, and they're shooting, 2,000 antibodies a second. Who are they shooting first? You see? The big guys, big mamas, tying things up, IgMs. Okay? Can you see here, the titer goes up. If you're a sicko, your titers should suddenly have antibodies, and then the levels should go up and up and up and up while you're fighting the disease. Okay, when they come down, that means hopefully hopefully you're not dead. Hopefully you're winning, right? So you don't need them anymore, so the titer's going to be going down. Check out who's in that titer. Can you see who's going to be shot first? Can you see the IgMs first? And then, of course, you're going to have IgGs as well. But overall, that means your titer's going up. For later, can you see we've got, what do we call them, memory B cells? Memory B cells, what do they shoot the most? Remember, it's 80% of your typical titer are IgGs. Can you see there? So you always have a stock of memory B cells that can shoot within how long? Well, let's take a look. Can you see there's a time? Hopefully you won, right? Disease agent is gone, expelled, you're better, you're recovering. Maybe 10 years later, this varies. Could be next week, could be 10 years later, it comes in again. Pathogen gets in again. Your body's like, oh, we know this one. Oh, within hours. Within hours. Check it out. Look at the difference in the size. You don't even know you've been invaded. That's how good your immune system is. So, this is the second, and if it happens again, third, fourth, fifth, you are pre-warned, pre-armed, ain't nothing gonna kill you now. Why do we vaccinate? We vaccinate so we can give you a harmless version of the pathogen that hopefully will do what? Produce memory, and then you are ready. You're less likely to be hospitalized. Even if you get a slightly different strain, it should be a little bit easier to take care of the business. Do you see that? So this is cool. But this is true for real infections as well. Typically, if you've had that particular strain once, you shouldn't have that strain again in your lifetime. Unless, of course, you're immune compromised, in which case you may not have that. Okay, so does that make sense? That whole response, and then memory. Very good. Okay, so does it take time to become seropositive? Sero, remember serum? That's the liquid portion of your blood that doesn't have any blood cells in it. It only has things like antibodies. And so it takes a while to seroconvert from being what? Negative to now antibodies appearing. Remember the graph? And then they go up and up. So what happens if you're seropositive? That means you have antibodies, yeah? But it takes a while. Titer, can I just review? Titer means how many? The number of antibodies. So what did we say happens to a titer if you're a sicko? They should be going up day by day by day by day until hopefully you're better. And then we can detect antibodies by their function. And I want to remind you that in lab, we actually used antibodies to do diagnosis. So there'll be more about this in Unit 4 when we talk about diagnosis. But can you see some antibodies are super good? Remember the IgMs at, what's that clumping process called? Agglutination. And some are really good at, can you see that process? What's it called? Opsonization. Some are really good at getting rid of the poisons. Remember what we call that? Neutralizers or neutralization. And some are really good at getting the knifing agents going, you know, that complement. So you get those MAC attacks. So they all have functions. What did we say, IgMs? They're really good at doing the A word, right? Like aggregating, agglutinating, whatever A word. Clumping, in other words. Use it for blood typing. All right. So, functions. This is a list, same list again. Same list again. Can you explain the first two? The first two are the same thing. They both are to do with the clumping business. What's the difference? Well, I hope you've had chemistry where you added at certain times a liquid to a liquid. So if you have a liquid antigen and you add it to your antibodies, which remember are liquid in your liquid blood, you get a solid. Remember in chemistry we called that a precipitate, do you remember? So if it's a liquid that's being turned into a solid, we call it more specifically precipitants or precipitation. If it's a solid, then we're going from a solid to a big clump of solid, yeah? So what do we call that? Agglutination, agglutinins. But honestly, it's the same thing, right? Clumping, okay? Clumping. I got pictures of that too.
Ooh.
Okay. We talked about opsonization. What does that mean? I hope everybody knows opsonins are good at coating, right? Remember, coating? What's neutralization mean? That means you're good at doing what?
Removing toxins.
Getting rid of the toxins. And so did you know that neutralizing antibodies are known as,
Antitoxins.
see? So I know you've heard of those. Oh, yeah, Jay has been bitten by a rattlesnake. Oh, we gotta get the antitoxin. Well, now you know what it is. It's antibody. You're gonna give Jay some antibodies. I mean, in my area, it's dogs get bitten all the time by rattlesnakes. So the vet, you have to take a picture of the snake. If it's a Western diamond rattle, you know, Western diamond bat, take a picture in, and then they can give your doggy it and hopefully save the life of your doggy. So, antitoxins everywhere. And by the way, one of the best areas for poison control through antitoxins, Loma Linda. They even made a show about it. I forget what it was called. It was called something like Poison ER. And they had Dr. Sean, who was always giving everybody antitoxins just around the corner. It was on TV for a long time. Yes. We actually had his best friend teach here, who's a herpetologist, snakes. He taught zoology for us and anatomy. Yeah, oh yeah. We got good people in the Inland Empire. Did we say some are good at getting complement going? We did. And now the last one you can ignore now, but we're going to come back to it. We're going to talk about how we're so smart in microbiology that we can modify antibodies. We can actually make them in the laboratory, modify them so that we can use them for detection. In other words, diagnosis. So we'll be talking about immunofluorescence, ELISA, that's coming up in diagnosis, all right? But those are special antibodies, so we're not going to focus on that today. So, summary, clumping, coating, poison control, knifing. I'm always somebody who goes, one word definitions that means something to me. You come up with your own term. But this is kind of a pivot slide where you can explain what antibodies do. Are we all good? Right? Are they effective defense, you think? Are they good for keeping you healthy? Does it take a lot of energy to produce antibodies?
Probably.
Yeah. Okay, so again, precipitants and agglutinins, I've got trap and tie up. Opsonization, this is the same thing again, third time through. Here we go, do I repeat myself? Oh yeah. Can you see, third time through. Coating, we had clumping. And can you see now the sticky out bits where, yeah, we've got, there you go. So now, what can we do? We can do that. Can you see, they're going in, and then, death, right? Opsonization. Awesome. Then we can look at our, like if you have toxins, you put it into, say, a little mouse, where's it going to go? It's going to go straight to the cells and destroy the mouse or the human, right? But if you add in the antitoxin, now we know what antitoxin is, right? They're the specific antibodies that bond to poisons. They will bond to the poisons, so that means they're out for the count, which means now, can you see, they've been blocked from hitting your nerve cells or your neuromuscular junctions or whatever it does. So isn't that cool? And we can actually give antitoxins to patients. It's like, oh my God, they're going to die in an hour. Let's give them the antitoxin, like the snake venom or botulinum toxin or diphtheria. Oh, diphtheria toxins are terrible. They are real killers. We used to lose a lot of babies to that disease because of the toxin. So now we've got ways to, yes, great. You know how some things can clump your blood? We can block the blood clumping. Yay, that's great. Use antibodies. Woohoo. And then my favorite one of all, which shows a blowing up cell. I'd love it to show you blown up cells. Can you see the knees? See it? The knees? Binds at the knees. You see, we get the first one, C1, and then that just cascades everybody else. Oh, wake up, wake up, wake up, wake up. Yeah. I'm going to teach by just noises now. Okay, so what have we just been talking about? Repetition from the beginning, everybody. From the beginning. You're born. Right? You're born. But before you're even born, you have every lymphocyte you ever need, right? Every lymphocyte you ever need. And they came from what? Your stem cells? And if we're talking humoral, B cells, where did they differentiate? Bone marrow, right? Bone marrow. How come they're all different? It's called gene, it begins with an R. Genetic rearrangement or recombination or shuffling, anything that means to what? Move genes around so that you get slightly different tips on each type of receptor, yeah? Have we got to get rid of the ones that match your heart and your lungs and your brain? Do you want to keep the ones that kill you? Or do you want to get rid of them? What do we call getting rid of them? It's like that button you push. Oh, deletion, right? Delete, 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 right? So we call them, remember they're called clones? Clonal deletion. Dum, dum, da, dum, dum, da, dum, right? Get rid of those clones. And so now there they all are. Where are they? Well, they're in your lymphatic tissues. They migrate. They're ready. You're born. You're born. Okay, the first year's a bit dicey because nothing works right yet. But after that first year, you should have everything together and you should be able to start mounting defenses, right? So what do? We've got to find the right clones, right? Matchy, matchy, matchy. So first we've got to do the, remember the phagocytosis and the presenting if you want a really good B cell reaction. They'll do it on their own, but it's like, meh. A really good one, we've got to do clonal, and the word is selection. Now you've got to select. I'll have you and you and you and you and you and you. All right, go forth and proliferate, right? So what gets them going? Interleukin 1. Remember the MHC2 presenting? Right? So then we've got what? Helper Ts. We've got interleukins being produced, 1 and 2. And the right B cell is plugging in as well. So the helper T is the right receptor. There's tons of them, don't worry. Same process, shuffling. We've got the right B cells, one at a time, because you've got all these presenting cells going to your lymph nodes going, okay, here I am, match me. And then somebody else is going, here I am, match me, match me, match me, match me. And then we have mitosis, right? Mitosis, proliferation. Then, right, we've got the plasma B cells. What do they shoot first? IgMs. IgGs are on the surface, getting everybody going. They're the ones who kind of interacted a bit. Then what have we got? IgGs, right, up to that. And then, you know, there's, you win. Yay! I won! GG. So now, got the memory, right, the second time. Now, you can't give somebody else your cells. Why not? Well, because you all have markers. Remember, you have MHCs and other markers unique to you. If I were to give you my cells, you would coagulate because your whole body would try and reject it, okay? But what I can give you is my antibodies because I'm human and I produce human antibodies. So I can give you antibodies, and that means you don't have to make them. If you've got a disease and I want you to survive and nothing is working, okay, I'll give you some of my antibodies, and that will save your life. And what do we call that? We can pass it on to you passively, right? But cells, no. Why not? They're too individual. They'll cause more harm than good. Okay? So, do we give them? Yeah, we give, remember, antitoxins. Aren't they antibodies? Sure, we do it all the time. Are we all cool with this? Now, when we want a B cell response, remember it's called, what is it called? Humoral. It happens at exactly the same time as the innate, but it's slow. It's slow. What else happens at exactly the same time? The cell-mediated. It's just I'm going to teach it to you one slide at a time. But we're going to go through T-cell mediated. And don't forget, in order to get a B cell response, we've got to have at least some of the T cells helping. What do we call them? Helper Ts, right? Okay. Righto. So let's talk cell mediated next. How's it the same? How's it different? Very similar. Kind of nice. Okay, cell mediated means we're not shooting anybody anymore. In order for a T cell response to kill things and to get rid of things, it's very much a touch. And T cells are kind of interesting because as soon as I touch, if I'm a T cell, if I touch you, you're dead. And that's how it works. So cell to cell. Now, typically, you're not a pathogen. You're an invaded human cell, right? By viruses, or you could be, I don't think it's mostly, you could be a cancer cell, or you could be anything like that, right? That has interesting markers. But as soon as I'm like, oh, can I touch you? Die. So it's very effective killing machine. It's all about killing, okay? And what's really interesting is if I touch you, you get holes. You literally get holes in you, and then I shoot enzymes in and disintegrate. It's very much cell and cell. Okay, so do you see when I said T cells directly interact with those foreign or those, funky cells that are ours that aren't working the way they're supposed to? So it's a touchy thing. So they're very good at eliminating what we call intracellular pathogens. Remember, viruses are the best example of intracellular pathogens. And so that would be what? Infected host cells. Yeah.
So that means that, like viruses and things like that, the T cells will only attack it once it enters the cell, not before.
Correct. Because T cells are different. We're not talking B cells now. But will phagocytes attack them? Yes. And that would be more like a B cell or a dendritic cell, or maybe even neutrophils, right?
So do B cells not attack when it's inside the cell?
Do B cells attack you? No, not so much. They're more like going after the actual campus. Yeah, but it's all helped by helper Ts and presenting, so they work together. So they are different. So when you go back to the beginning again, look again at our overview of how are B cells working differently than T cells, and you'll see confirmation of what you just said. So yes, that's right. Good job. So the last C word is cancer cells. Remember, they will get rid of cancer cells. Unfortunately, there's a dark side because sometimes, when people have major illnesses, we might need to give them a new liver or a new kidney. And unfortunately, no matter how good a match we have, it's never perfect. And since T cells are responsible for looking at host cells that aren't right, it's the T cells that destroy transplants. And so if you have a transplant, it's a matter of time. We're always worried about the T cell response because that's why, if you heard of rejection, like tissue rejection or transplant rejection, it's these T cells. So don't worry, we have medications for that, but We'll get there next unit. Okay, so what are you putting in there? Transplanted tissue. They will, but the closer match we can get, the milder the rejection. So that's what we try. They work. Okay, the other thing to know about T cells is that, yeah, they'll make you collapse, but they don't do the cleanup. It's the macrophages that really take care of the business. So they'll kind of get everything going, touch it, and then the macrophage will then take over and will kill anything that's left, and then we can, phagocytose and get rid of everything, okay? Now, when you get the T cells are activated, you are again going to have this whole process of antigen presenting, helper Ts, but this time instead of B clicking in, we're going to see matching T cell clicking in. And T cells come in different flavors, but they are all cell mediated. It's all cell by cell. You can't give cells to anyone, remember, you can't pass it on. So we've already come across one type. Remember what were they called, helper T cells? Now I have to pre-warn you, immunity is a complicated process, and helper T cells, there's multiple types, but we're not going to go into that much detail. We're just going to call them all helper Ts, but you'll see helper T1, helper T2. Don't worry about it. They're all kind of helping with the process. Cytotoxic cells, they're the ones, they touch you, your cell dies. So why? Toxic to cells, and that would be to our cells mainly. They will sometimes tackle a pathogen, but in general, it's going to be our host cells that have been invaded or that are not working right. And so we call them TC cells, okay? And they have different markers on them than the helper. Often we call these CD4s and these CD8s, but that's a very typical name for them. And just like B cells, do you want to be equipped for future? Do you want to be hit again by that same pathogen? No. So what do you have also is you will then stockpile what we call memory T cells, and they'll be ready to move immediately if the pathogen ever comes back. Okay. So how does this whole process occur, and what does a T cell receptor look like? Well, can I review B cell receptors? B cell receptors were how many chains? 4 chains. Remember the 2 heavy and the 2 light with the disulfide. Remember, they can either be receptors, stationary, or you can actually shoot them out by protein synthesis. T cells are different. They never, ever are free. They can never be shot out. They are always tied in place. So can you see, this is a T cell, and can you see the T cell plasma membrane? You see the bilayer here? This is going around an entire T cell. And what is sticking out? Can you see this cup-shaped little receptor? You see one polypeptide here and a mirror image polypeptide here. So how many chains? Not 4 this time, but 2. Okay. So how come this receptor is going to be different on this T cell from all the other million T cells? Because remember, you're born with all the T cells you'll ever need, just like the B cells. Same process. It's called genetic shuffling, genetic recombination, genetic, the whole business of